Well, it's a real honor to be asked to give the epigenetics or epigenomics talk, and I hope to convince you that there have been major breakthroughs in the understanding of epigenetics and its relevance to cancer, surprisingly from the efforts of the Cancer Genome Atlas kinds of projects that we've been hearing about. So I'm going to describe the cancer epigenome to you. I'm going to focus on the role of chromatin. Just to remind you where this word comes from, it comes from Walter Fleming, a German biologist who in 1879 found that the nuclei of cells stained very strongly and christened this stuff chromatin. And of course, what was discovered very early on was that in cancer, chromatin gets significantly disorganized. And in fact, if you get cancer, one of the first things that's going to happen is a pathologist is going to stain a piece of your tissue with a dye and look at the chromatin. We've never really understood why the chromatin was becoming disorganized. And I think we have some interesting hints now which might suggest why this is the case. So I want to remind you, this is a picture of a chromosome at the bottom here. The DNA does not occur as a purified molecule in cells. Rather, it is wrapped around these beautiful structures called nucleosomes, which, by the way, are about 500 times smaller than a nucleus. And if you look at detail at the transcription start site, you'll see that these nucleosomes have tails of histones which are sticking out. And these have a variety of covalent marks which are applied to them, which have information coding potential, which specify how genes or which genes should be expressed. There are also covalent marks which occur on the DNA molecule itself. A certain percentage of cytosine residues become modified. This is called DNA methylation. And this has important information in terms of specifying which genes are going to be expressed. Now, a key is that these covalent marks communicate to little nanomolecular machines that move nucleosomes around to expose pieces of DNA so the transcription can be initiated. So the exposure of the DNA is really critical, which is why I thought this banner was rather timely, exposed the DNA dilemma. So if you want to know how genes are regulated, it's all to do with exposure. It's exposing the DNA to transcription factors and to regulatory molecules. So finding out where these regions are is really critical. Now, normally the way people do this is to use a nuclease to chop up that DNA which is accessible to an enzyme. And we thought this was one way of doing it, but maybe another method would be to use an enzyme, a DNA methyltransferase extracted from a bacteria, which can find regions of DNA which are exposed and put methyl groups on them, a covalent mark, but cannot do this when there's a nucleosome there. So we're interested in knowing what's the relationship between DNA methylation and nucleosomal distribution, because we think this is really critical for deciding which genes are going to be expressed within a given cell. Now, in order to find these chemical marks, which either have been put there on the cell itself or by this enzyme that we've added to the nuclei for 15 minutes before we extracted the DNA, we use a technique called bisulfite sequencing, which I'm not going to go through in any detail. I just want to say that this methodology, a chemical method, allows us to see the chemical marks. And so what we can do is we can tell which sequences, which cytosine residues, which were actually modified by the cell itself, by DNA methylation. So the cell, your cells, have 3 DNA methyltransferases in them, and you methylate a certain number of the cytosines which are upstream of the guanines. You do not methylate cytosines which are downstream of the guanines. But this particular enzyme actually likes these ones, and it puts the methyl group here. Now, there is a slight problem because some sequences could be sequences of recognition for either enzyme. In other words, a GCG could be recognized either by your enzyme or by the bacterial enzyme. And so we can't really look at those, but what we do is we remove them informatically. So now what we can do is we can find out which regions of DNA are exposed or accessible, and which regions of DNA were methylated in the human cell, and see if we can find out what's going on during the formation of a cancer. So this sequence, this procedure which we call GNOME-seq, which is nucleosome occupancy and methylation sequencing, works like this. Again, I don't want to spend too much time on this, but this shows you a DNA molecule in a cell. The pink guys are nucleosomes. The lollipops which are pointing upwards are black, have been methylated cytosines by your own DNA methyltransferases. These are GC sites, which are down below here. When we add this enzyme, we make these ones teal here, which we can see now because they become accessible, and the ones which are wrapped around nucleosomes are inaccessible. So this very easily allows us to see where the nucleosomes are and where the DNA was methylated in the cell. And then we can either clone individual molecules, look at an individual promoter at one time, or just put the whole thing in a sequencing machine and figure out the relationships between these two critical processes. And then we can detect these regions which are called NDRs, which are nucleosome depleted regions, which I believe are really critical to the function of the cell. So this just shows you how we can do this by cloning individual molecules. You're looking here at a DNA repair gene. You're looking at the transcription start site, which is where the arrow is. And these are human fibroblasts, which are actively expressing the gene. There is no DNA methylation, very few black circles here. Very accessible, lots of teal. Each one is an individual molecule. And this beautiful pink area here, downstream of the start site, is where there is a nucleosome position just downstream where transcription starts. That's called the +1 nucleosome. And in a colon cancer cell line, this has become totally methylated by the DNA methylation machinery within the cancer cell. So the DNA repair capability has been shut down, but you can see now that there's no accessibility, there's no exposure of the DNA, the bacterial enzyme cannot access it, and the whole gene, the repair gene, is shut down, and this person would have colorectal cancer because they can't repair their DNA. So we've just recently done this genome-wide. I want to just show you some of that data. We just recently published that. Instead of just looking at one gene at a time, what we're trying to do now is to go and look at all of the genome at one time. What is the relationship between DNA methylation and DNA exposure? Some interesting results. For example, if we look at sites, which are binding sites for a thing called CTCF, which is called an insulator, which actually anchors DNA sequences, and looked at the distribution of accessibility, the teal color again, and the endogenous DNA methylation, again, which is shown in the black color, you can see this absolutely remarkable curve here, where if we center everything on the basis of where the CTCF sites are, there is no DNA methylation, the black one goes right down here, and no nucleosomes in this region. And as we move out, these nucleosomes are beautifully positioned next to this anchor site, but that the DNA methylation and the accessibility run counter to each other. You can clearly see these two peaks touch each other throughout this. And so, just to put it in a more simple fashion, the DNA is known to be anchored at these insulator sites. That's why I've drawn this as an anchor. And the nucleosomes, which are on either side of it, are actually beautifully positioned as indicated here. And from that analysis I just showed you, which is genome-wide, looking at this distribution, we can see that the methylation, again, I'm showing this in a black circle here, is slap bang in between the nucleosomes. So the region between the nucleosomes is where the cell is methylating the DNA. And so this is really interesting because this anchor is obviously important in the organization of chromatin within the nucleus. And so maybe disruptions in this could actually contribute to the cancer phenotype. Now, we've known for many years that the DNA methylation patterns of genes are profoundly influenced and altered in cancer. This shows you a cartoon. I'm just showing you one cartoon here, but there are many, many cases in which this has been found. In normal cells, a gene which might be expressed has a rich region of CPG sites at the start site. Those are the open lollipops pointing upwards. And the gene is expressed, let's say it's the P16 gene or the MLH gene, and this would downregulate the growth rate of cells. Now, it's been found that in cancer, in addition to mutating genes, in addition to losing genes, the promoters can become inappropriately methylated, and the gene becomes silenced. And you can imagine that this would increase, for example, the growth rate of cells. Now, what I showed you earlier on is quite clearly with the gene MLH1 that this DNA methylation is strongly associated with a loss of accessibility of the promoter to transcription factors. So we're beginning to be able to relate these two processes together and to understand that they could be important for cancer development. Now, there's one very important thing about the slide that I'm showing here. This gene is off, but it's not mutated. And so the possibility is that we could reactivate it, turn it back on again, and make the gene silenced, make the gene work again. And I'll finish my talk with a description of how we're trying to do that with epigenetic therapy. You'll also notice that I have drawn in the coding region of the gene, sites which were methylated have now become demethylated. So there really is, it's like a seesaw. The methylation pattern is changing. So sites in the genome which were methylated before become less methylated in cancer, and there's a switch for methylation of these promoter CPG islands. Now, so far we've been doing this sort of one at a time, and now we try to look at this more globally using the assay which I described to you, the GNOME-seq assay, which allows us to interrogate where is the DNA accessible, and at the same time as we could look at where the DNA is methylated. And in our initial experiments, what we've done is to compare normal human mammary epithelial cells, which can be cultured, and we've compared those to a cell line called MCF7, which has been in culture a long time. It's not a perfect model, but it was derived from someone that had breast cancer. And so the question is, what happens on a global scale to DNA methylation and to accessibility of these regions as a function of transformation? So this just shows you globally what you can see if you look at the level of methylation of CPG sites. That's the normal methylation in a normal human mammary epithelial cell. You can see that overall the level of methylation has decreased in the mammary carcinoma. So as I said, there's a seesaw, there's a change in distribution, there's less methylation, but more focal methylation. And you can also see that generally speaking, there's not much change in accessibility. About 10% of your chromatin is accessible. The 90% is closed down, but about 10% is accessible. And this doesn't really seem to change much in cancer. However, the distribution and the positions of where that accessibility is changed is actually fairly dramatic. So here we've actually compared the NDR, remember this means nucleosome depleted region or accessible region, between these two cell types. And I still have trouble believing this result, and this is a very early result. I hope it pans out. But it's extremely interesting to me that there is a massive change in the distribution of nucleosome depleted regions between these two cell types. In fact, you can see that in the mammary epithelium, in the cancer cell and the normal counterpart, only about 13,000 nucleosome depleted regions overlap. So these regions have really switched around a lot. And if you look at more detail, in the right-hand side, you can see that CTCF sites become more prominent in the MCF7 cell, so more regions becoming anchored in the cancer cells. At the same time as it looks like many enhancers, which also have nucleosome depleted regions, become less frequent. So this is very early days, but we think that this is actually sort of interesting in terms of describing the extent of what happens. It's not the overall amount that matters, but the distribution is very, very significantly different.